All right. So everybody, welcome to kind of the pre-record here. Again, these are mainly for my faculty evaluations, but in an emergency, you could use this as a sort of backup plan, if your Zoom blows up or something like that. So we are covering lab 3 this time. This is action potential. So this is going to look at neurons and how they essentially fire, as I've called it. This is how they're going to communicate. In these little channels, as you can tell, this is the neuron in green. That's where the cell's nucleus is. And remember, neurons are just like every other cell. They're just shaped super differently because all these axons are going to go and connect to different neurons. And that's really, like we talked about with the brain, it's not that there are more cells that show up. It's that more of these connections start showing up. And I'm not 100% certain on this image, but I think the red in here is the sodium channels, and we're going to cover those. And remember, follow the sodium to see where your action potential is firing. And the sodium ions themselves, they're the ones that provide a lot of that charge. We're going to see the potassium role that kind of comes into play, but ultimately that's my basic thing. So cool. This is the human brain. There's a lot of components. This is not a neurology class, unfortunately. I think that science is really cool, but for the most part, and we're actually in lab today going to be actually doing motor neurons, which are connected and can be dictated by the brain, but they actually span your body and they're still going to operate the same way. But for the most part, what makes you human and the unique and like all the innovation, thought, strategy, making up things that don't technically empirically exist in the world, and it's a very important human thing. Think about money, it doesn't have actually any actual value, for example. That happens in the neocortex. That's why we have such a huge head, is that this frontal area is thick with connections. And when you're born, you have that number of cells, but as you get older and quote unquote wiser, this area builds more connections. Cerebellum, so thalamus is actually going to be very important for voluntary action. So people that lose function of their thalamus a lot of the time can still think, but they can't act on it, for example. And that's a big connection to what we're doing today with the motor neurons. So that's actually very interesting. When you see midbrain and hindbrain, these unfortunately sometimes are going to get characterized as lizard brain because reptiles that are pretty basic still share a system that pretty much looks like this. Lizard brain is an oversimplification, I'd say, but you don't have, you can kind of take a look at those and see what your interpretation is of each of these areas. So I definitely recommend looking into a lot of these areas. Are really unique and cool. But again, we're here to focus on one neuron connecting to another neuron or many others. So just like we covered in that first lecture, you have cells that are responsible for interpreting the signals from your environment. So if we're looking at this, these are going to be your ear cells and some of those, some of the vibration cells that go in when sound waves hit. That sensory input is going to be sent to the brain, though. The brain is the integrator. Everything has to be interpreted as a single signal for us. The brain does that. It takes all the information that we got from these cells over here and turns it into a signal that we can act on. And this is a very good image right here, because ultimately the brain in a lot of cases is going to take an action. For example, in this case, if you hear something dangerous behind you, you will start running because that danger signal will come in through your ears. Your brain will integrate that danger. The reaction will happen and you will have an effect because of what happened. So in this case, the brain isn't moving the muscles. The muscles are the things that move, but they are the things that fire to put them on and make them start heading the right direction, for example. So this is your biology of a neuron. It is a very unique looking thing, to say the least. You're going to end with, so to begin, you're going to have things called dendrites. These are going to receive signals from other neurons that are connected. The way neurons fire usually starts with them receiving a signal, then they fire. This is where you receive the initial stimulus, in this case, that causes the firing. Along what we call the axon right here, highlighted here, that's where the sodium is going to start like bursting in that pulse, which is the action potential. That's going to fire along down this line, and it's eventually going to hit at these synapses. See these like small, see how these are kind of touching the other neuron? This is going to be happening up here. This is what happened to the original neuron that we're talking about, but we can zoom in here, as you can tell, and this is where the sodium influx and that action potential causes the firing of these little vesicles that can contain neurotransmitters. These little neurotransmitters are what connect to the next cell, and we're going to start the process over again. Because remember, It's the firing of the connections that matters. It's the one neuron communicating with another neuron. That's how you get a pattern or a circuit starting to fire. So as we'll see, neurotransmitters are one way that you can communicate. You can also directly pass on this sodium signal that's coming from cell number 1 here to cell number 2. That's more of a quicker reaction. Think about why, like when you make a knee-jerk reaction, for example, you're not really emotional about it. But if you have a more like emotional buildup towards it and you actually have like a feeling in your body, that's when neurotransmitters have usually taken effect, because they can actually seize a lot more. They can actually usually seize at least a lot more of the effects. They're a little more powerful, and we'll see why. So, lots happening in this slide. There's a lot of options for what you want to follow in this case. So don't let it intimidate you. Like I said, the easiest way it's always been for me is to follow the sodium. So if we start, and what we're looking at, we are looking at sodium, and it is huge outside the cell. That's what these little brackets are. Remember, that's the concentration. It is really high outside the cell. So look at this. It's about 90% outside here and only 15% here. Or sorry, only 10% here. I don't know why I said that. So there's a ton of sodium out here. And because of that, you have tons of positive charges out here. Because again, the sodium, like I said, if you follow where the sodium's at, you're usually going to find where the positive charge is at. So what's going to happen in these axon areas is that you are going to basically build up this neuron, this sodium charge, so that you can eventually let the sodium flood in. Because like we said, remember, there's two things that make a electrochemical gradient. There's tons of physical sodium molecules, and they want to push in. And all this positive charged sodium now wants to get inside the cell, which is negative charged, because you kicked out all the sodium. So it's actually that plus minus attraction too. So you have two forces working in. So this is your typical buildup outside and inside the cell. As you can see, you have the sodiums in yellow. I ruined it by going over it. And you have tons of these little yellow guys out here. So you're building that big gradient. Okay. So we'll revisit this, but this is what you're going to be working with a lot today. And this is the stimulus. This is the action potential, as it's called. So we'll see this. And this whole thing is going to be for one cell, I should say. So remember that no matter what, we're working with a single cell in a lot of cases, or at least a single set of coordinated cells, as we will see with the frog leg being a coordinated motor neuron. So a couple things to point out. Where you start, where a neuron that is not firing, this is something called resting potential. As you can tell, it's about 75 millivolts right there. Negative 75 millivolts. That's because, remember, we're measuring the outside of the cell in this case. We're measuring the inside of the cell. See, it's hard. And the inside of the cell, remember, is really negative. Okay, so that's what we're measuring. When you fire a neuron, remember, you're going to let sodium in. That's why you see this big upswing of charge. And as you can tell here, you actually, inside the cell, you actually breach past zero charge, so neutral charge, and you actually take in a ton of sodium. And that's why, again, you have this big positive charge show up when the neuron fires. Once the neuron fires, it starts to kick the sodium back out, though. And this is through the exchange, again, with potassium. But ultimately, like I said, if you follow the sodium, keep getting kicked out, all of a sudden, the inside of the cell is going to be negative again, because you kicked out all those positive sodium ions. And again, right here, you're going to reset a little bit. So let's take a look at this system in a full application. Maybe I should have started with this slide, but hopefully it's okay. So here you are in your resting state. As you can tell, you got tons of positive sodium charge out here. So we're here right at 1. I should move that. We're at resting. So if you get a stimulus, though, the first thing that happens is some of this sodium starts to flow in. Some of these channels allow the sodium in all of a sudden, and that's only going to happen if you have the stimulus. So a few of the channels are going to show up, and a few of these sodiums are going to make it inside. That's why we start seeing at 2 right here, a small bump towards the positive charge. See how this is going to be rising up on this scale towards a more positive charge. It's not past 0 or at a positive charge yet. This is the beginning. So during the rising phase, and this is 3, this is when the firing is really happening. Yes, I am definitely enjoying it, but go away. I don't know why that would show up. Okay, so at 3, Now everything is coming in. Now all these sodium channels in this at this part of the axon where the firing is happening, they are all coming in. They are flooding in super quick. And as you can see here, that's going to be positive charges are going to start building inside the cell. And that's why at this point, at the action potential, we're actually, somewhere around this point is like, plus 25 millivolts or something, right? You've actually turned the inside of the cell positive. You've completely reversed things. So what this does is that at that positive charge, the sodium channels actually start closing again. They close, and at this, with this positive charge coming in, all these potassium channels now open back. Now the potassium channels have finally entered the chat. They open, they allow their potassiums, noted here in orange, to go out again. And that's actually going to start rewiring everything back to being positive outside, negative inside. And that's why with 4 in green right here, you have that big fall of charge. You stop letting sodium in, and you actually let some potassium out. And so that is going to cause the negative charge of the inside of that neuron to return negative. So again, here in the final phase, basically, as you can tell right here with 5, you actually go a little too negative for a bit. And this is kind of just the reset phase. You can let a little potassium out until it's okay. And then eventually, once you get back to 1, because remember, this is all a big cycle, you're going to close those potassium channels again. So potassium has a role in resetting things, I would say. So I would call that, I think that's pretty sure that's called repolarization. Let's actually take a look, because again, the language is where you're going to have to practice a lot of this. So be sure about the language, and I'll kind of show you some of my tricks that I use, but be careful, because that's what you're going to see with a lot of exam questions is this hyperpolarizing, is this depolarizing, etc. So we can match where the ions are going with that language and hopefully set you up really well for that. Okay. What this is showing is, all right, this is why the action potential is something that travels. The action potential, again, is just the sodium that is influxing in, okay? But that is happening at specific spots on the neuron, okay? See how this is actually shifting over. What it's causing is that as the sodium shows up in one section, it causes this section to fire. As this section fires, it's going to cause this section to fire, and so on and so forth. In green, you can actually also see the key role, again, of potassium. It's going to start resetting the neuron back to its like happy, ready to fire state. You need potassium in this case to get back to that state. So although the really important firing and like the big plus charge coming in and like, really like ionizing and like hitting the cell to make it go, you always need to reset that. If you don't reset that, your neurons just won't work again, because to reset it means to get that sodium back ready to fire. Potassium's the way that we actually reset that. But as you can tell in this blue right here, that's what is actually happening, is you're actually following along the axon, and eventually you're going to hit the ends here. That's where you're going to fire your neurotransmitters or your signal again. And that's how you communicate and fill a pattern. In this case, what these channels are is something called voltage gated. They're only going to open and allow this whole up and down process of sodium if they feel this positive charge show up. And that's what's going to allow the channels over here to allow the sodium in, and so on and so forth. So it's kind of a sequential thing. I kind of think of it kind of like a pulse, but it's so fast that it looks just like an even signal, basically. So again, this is always going to be my trick. Where is the sodium going? And you should be able to find where the positive charges are going. So this is like kind of the big picture. This is like the big slide. This is what I'll obviously post, I would say, as a PDF probably, so you can kind of view it. When you're seeing that firing, this is what you're seeing. When you're seeing that resetting, this is what you're seeing. So this is where we can start seeing the the language really being applied. So in that early set, actually we can do it down here. So in this early stage, so this is where you're at your resting, you get a stimulus though, this is depolarization. So in the resting state, you are polarized because you are not at 0. Okay, that's what that means, is you're polarized. Remember, polar is like opposite, it's like, or like two different types of things. Polar means like different. Basically, you are in You are not even, you are different. These neurons are negatively charged, remember. So here, I'm actually going to erase these to start over. So at this stage, you are polarized. When you start letting the sodium in, you are depolarizing, actually, because you are reaching zero. To depolarize means to even back out. And you are in the business of the direction of evening back towards zero in this case, because you are going away from that negative charge you had established. Up here, you actually burst past the zero here, so you actually polarize again. But usually you're not going to see language that describes that. You're just going to see this peak right here. And this again is where the potassiums are going to reset the charge again. What this is called, so after you have depolarized, so you've gotten closer to zero again, you actually have to return, and that is repolarization. That is where you are going to return the neuron state back to its negative state. And then again, like we said, you kind of go a little too far back, and you actually have to do what's called hyperpolarize again. And you actually, again, hyperpolarize means stay polarized essentially, or get back to polarized state, which is going to be that negative again. And you want to hyperpolarize to reset and have your channel ready to fire again. Okay. So a lot of us in this class are actually really interested in the neuromuscular system and muscle systems. The key way that your voluntary muscles are going to fire is that they communicate with a neuron. The thing that we're looking at today, the frog leg, is a muscle, is the neurons paired to muscle, for example. They are going to tell and send that signal to allow the muscle to fire. The thing that they actually send, in this case through the synapse right here, is a neurotransmitter that is going to allow calcium to flow in, and that calcium flowing in allows the muscle to contract and basically have an action all of a sudden. So anytime you have that, it is a neuron, a motor neuron in this case, telling the muscle to fire. And that's where you get your voluntary actions. So interfering with this process, or maybe even trying to improve this process, is a very neuromuscular field of science. You have to understand both of these systems so that you can change it or fix it or improve it, for example. So this is something we're also going to deal with. This is experiment 3 in this lab. What are the refractory periods? All these are if you have 2 stimuli. So remember, we've dealt with 1 stimuli so far, and that's kind of what we're seeing here. If you have 2 stimuli, what happens and how quick can they be? So in the absolute refractory period, when you're still firing all these sodiums, potassiums, and stuff, any stimulus in this entire period right here, in this absolute area, nothing's going to happen again. The neuron's already firing. You can't fire it again. That's like trying to move twice. You're not quick enough to, basically, right? And that's because your neurons have not reset yet. They're not ready yet. So in the absolute, you're not going to get a single movement from 2 stimuli. So if you try and, so we stimulated right here, and if we tried to send another stimuli right here, nothing's going to happen. There's not going to be any cap, as the acronym's called, and you'll read that in your lab book. There's not going to be another action potential that shows up. So this week's lab, we're not actually going to measure relative, but it's good to know it because, it might be on the exam, right? What relative means is that if you get a stimulus after this big peak, and you're kind of right, you're in this stage, you're probably going to get a small action potential to be like, oh, boop, like one more again. Actually, it's probably going to be like small, like really small like that. So what that means is you're not going to be able to re-engage all the neurons in this phase. You're going to engage some, but you're not going to engage a ton. So it's going to be a really small blip. So past the relative period, though, if you send in another stimulus right about here, so you remember we're back to this stage right here, you are actually going to receive the full action potential one more time. So then you're out of that refractory period and you're good to go again. So think about athletes when they have to make really quick movements, for example. This is why quickness is inherently something different. You can train your neurons and your neuromuscular junctions to be a little quicker sometimes through, a lot of the training techniques that we champion in sports, but there's a certain limit where the neuron just can't fire again. It's already firing, right? So it's actually pretty cool. All right, so we'll do this in class. This is, again, the gaps. Here are all these good questions here. Keep these in mind mainly for the exam. I'd say you probably don't need to come in ready to hit all of these during lab because you're going to learn about a lot of abnormalities in that case. So I wouldn't worry. So let's cut into some of the brain chemistry, because a lot of us will deal with neurobiology in some sort of sense again. So again, brain doesn't gain mass. Even when you're a newborn, 1 month, 6 months, you can tell they're not really changing. You're not really multiplying more cells. You can have more neurons show up. That's fine. They don't get, to get smarter and bigger and more wise and have more information theoretically in your head. Your head doesn't get literally bigger, especially once you're not growing as a small kid anymore. It gets thick though with more connections. Remember those dendrites and those synapses and stuff? This just gets way more interconnected because it's not just one neuron goes to one thing. It's that one neuron can make as many as 100 or 1,000 different connections to other neurons. That's where you get really complex. And that's why the human brain, despite being, it's pretty big, but it's not huge enough. It contains somewhere between 100 and 1,000 trillion connections between neurons. And the connections and the thicket of connections, not the number of cells, that's the key to the complexity. So we've seen this before. Individual neurons can pair together into these small patterns called circuits that fire together, and then those can start communicating with each other, and that's what forms some really cool patterns. And that's kind of the neat thing in that case. So for an example, we can look at instinct essentially. Instinct is when you see something, smell something, hear something, and your body just reacts. Your brain knows that pattern immediately. On the bottom, you can tell humans, it's kind of a debate whether humans actually have instincts anymore. Probably one of the key instincts I would say most humans still have is that cry of a newborn will cause any human to usually go into a more frantic and emergency state, because the cry of a newborn should indicate something is in trouble. A newborn's in trouble. We're going to go find it. But you can actually get a lot more complex and really interesting when you think about visual instincts. Why are certain animals, for example, scared of other certain animals the minute they're born? They don't have to learn to be scared. Humans, we usually have to learn things, which is a good advantage in a new environment every time. But a lot of animals exist in environments that don't change that much. So, for example, zebras are really, really afraid of humans. It kind of goes into the domestication theory. Why did we not domesticate zebras as Homo sapiens? Why did we go out into the Asian steppes, find horses, which are relatives to zebras, and they were able to domesticate them? The reason is that zebras just hate bipedal primates, us. They hate us. And how do they hate us? And if you visualize this, there are certain circuits that can create images in the brain that can come sometimes as a blueprint. So there are certain circuits that do straight lines, curved lines, what's the background, and what's the color. So in the case, I'm actually going to try and do black here. Okay, so just to do a stick figure. If a zebra brain is going to show up and it's going to see human, first thing is that the neural circuits can see shapes that are straight. So we can call this, you can probably see straight legs, you can see straight arms, and then the curved circuit is going to see this straight head up here. Here's a human. It's a pretty bad stick figure I'm drawing with a mouse right here. And then the background. And the background is going to allow you to differentiate this shape. Color too. Color is actually pretty big with Homo sapiens and primates. That's a whole nother evolution story though. So when you see this image as a zebra, you're upset. And that's it. The combination of straight and curved lines put up against the background of your visual CNS is going to cause you to be unhappy. There's a reason behind this, is because Homo sapiens were a dominant predator throughout Africa. There's a reason that all the African mammals that exist today still exist, is that they evolved against us. They beat us in a lot of cases. And there's also a lot of reasons why they hate us in a lot of cases. So for example, we domesticated dogs once we emerged into the Fertile Crescent, into the Middle East and into Asia and into Europe. But African painted dogs still hate the sight of humans. And African painted dogs actually have a better pack structure than the idea of the gray wolf. They're actually a lot more caring, a lot more social, empathetic. They're actually better candidates for domestication than the gray wolf theoretically was, but they evolved next to us. And they evolved next to a bipedal primate that was very, very dangerous. And so that's why, to this day, everything in Africa is still so dangerous. It's because they evolved to beat us, to live alongside us, which means you have to be pretty dang dangerous. Pretty cool story. Sorry, you could have sped past that probably. But the way that you have these complex interactions and these complex circuits is that this is one neuron right here. Look at these hundreds of synapses from other neurons attached to it, ready to fire whenever they have the chance. And any of these can fire this neuron right here that's being poked at right here. And this neuron is likely connected to 100 or maybe another 1,000 other neurons. And that's why the brain is so complex. And that's why when we try to design AI, for example, that can weave and move between these connections and decide and prune and change everything, it's a very difficult process. So again, this is what we're looking at. This is our single neuron right here. This is the motor neuron that we're going to look at, which is really cool because it's really long and that axon has to be huge to communicate that signal down to the muscles. But this, the inner neurons, this is that thick neocortex like I talked about with humans. This is where the complexity of who you are, your decisions, your memories, that's where they all exist, is these connections. And it is that thick and that crazy. Like I said, theorize that you have 1 quadrillion, so 1,000 trillion connections between your neurons. And that's why we can be so unique and complex. So there's 2 options here at the synapse. One is that we actually just continue firing that sodium channel. So you don't always need to exchange neurotransmitters. You can actually just fire the next neuron to fire again. You know, sodium for sodium, fire, really quick. Neurotransmitters are actually a little slower in this case because they are actual big molecules that will attach to certain cells, neurons, and cause them to fire. They're a little more specific and they have a tighter grip, basically. So here's a small example of how to assess that in yourself. This is the trolley problem. I'll let you read this right here. What this problem is going to ask you to do is engage the logical side of your brain. The logical side of your brain is going to say, hey, 1 person is better than 5, as you can tell here. Most people are going to choose to pull that train, basically. And they're going to pull this. We'll lose 1 person instead of 5, right? And that's usually what you see, because that's a logical mathematical problem. Okay. Now the problem is the emotional reverb of this problem is to stop the train instead of pulling a lever, you actually have to push a person in front of the train to derail it. The train derails and saves 5 people down the track, but you literally pushed the person in the train into the train. You're engaging a very different side of your brain in that case. You're engaging an emotional side. And that's why most people can't bring themselves to push the person, because you're engaging neurotransmitters in that case, not just a logical step-by-step, this makes sense. No, you're engaging empathy. And empathy is one of humanity's biggest advantages, is our brain is built to connect, to be social, to partner. And that's why in a lot of cases, emotion is going to beat logic in a lot of human situations. Think about all the situations maybe you've been in or you've seen people be in where it's like, why did you do that? You made an emotional decision. And it's because neurotransmitters have a tighter grip on things. They cause more brain satisfaction, more brain pain. They can sear and strengthen neuron connections a whole lot more than logical, small electrical connections usually can. This is a left brain, right brain image. That's a oversimplification of what happens. I'll let you kind of read into that as you will, but there's a lot of emerging science on, the fact is that the brain can interconnect and decide where certain regions are going to fire. You don't actually have a place built in for each in a lot of cases. So what happens when a neurotransmitter fires is that first step is actually calcium. You don't have to know, I don't think you have to know the calcium part, but calcium's the final step in the neuron firing. It actually, instead of sodium, once it's allowed in, it bursts these little bubbles that are filled with the neurotransmitters. What happens at the next neuron is that the neurotransmitter is actually going to attach to a channel, and that's going to start the sodium influx. So to start the sodium influx with a neurotransmitter axon, you actually have to have that neurotransmitter attach and then allow the firing. And eventually, the neuron detaches, the firing stops. But they stay on a lot longer than just a strict connection, and that's why emotional responses feel deeper and last longer in a lot of cases. Here are 8 of our biggest friends in the in neurotransmitters. Each of these has different roles, and they're not as simple, as this slide can always make them. But in a lot of cases, those of you maybe that meditate, what you are releasing is GABA. That's actually calming you down. Serotonin is more of a general mood transmitter. This is something that keeps people kind of even keel. It's not necessarily happy or sad, but it's like we can stay normal right here. We can stay stable. Now, dopamine or endorphins like oxytocin, these are happy. These are big rewards. This is why when we eat sugar again, we get a big burst of these and our body's like, that was good, do it again, right? And with learning, there are different neurotransmitters that signal, we just learned something new. This is going to help us survive later. Burn this into the neurons by attaching these neurotransmitters to it, because these two, glutamate, acetylcholine, these are super duper like attachment heavy. So this attachment up here is going to be really strong with those. That's why learning, when you learn something or you have a memory, it's there for a long time. So it's kind of cool. One of the other things is that, esoteric drugs or even prescription drugs that you may assign in healthcare, these manipulate these connections. They can stop reuptake. They can cause reuptake of neurotransmitters. They can cause neurotransmitters to stick. Some drugs, in a lot of cases, literally just mimic the neurotransmitter and cause stuff. So others stop the removal of other ones. So like you can see right here, if you stop dopamine removal from the neurons, you stay in that elated state, for example. A lot of, prescription drugs, for example, make you reuptake serotonin so that your mood stays and you don't go up and down so much, and serotonin can help with that. Now, this is a very inexact science. In a lot of cases, it's kind of the paradox that we know these drugs work, we don't necessarily know 100% why yet. And that's kind of an evolving science as well. So, last slide almost, sorry, but the human brain's a very cool thing. We're going to be looking again at motor neurons today, but don't ignore what the CNS is capable of, what your own brain is capable of. It's very, very cool. It's an evolving, changing thing, and you can strengthen or lose different connections, for example, because your brain is always trying to make room for more, and you do have to get rid of stuff in a lot of cases. You do lose memories over the time. So brain is a very cool thing. I'd advise reading about it if you ever have the chance. It's very cool. I have books if you want, let me know. So one last thing. This is where brain genetics start coming in. For example, we discussed serotonin being around, and if you have serotonin, you usually are a more or less impulsive person if your serotonin is nice and even. In the case of people that are born without something called serotonin receptor 2B, and that's what the little line means, if you don't have that receptor for serotonin, serotonin basically falls on deaf ears sometimes. Sometimes it's released, but it's never read. These humans, in a lot of cases, do not have the same amount of serotonin activation that somebody with this gene will have. What this can cause, theoretically, and we have demonstrated it in mice, is that somebody without these two genes, or without this gene, will not be able to control their impulses as well. And that's where you start getting into some very interesting behavioral genetics. When you get into neurogenetics, you can start to see, okay, how is somebody's mind shaped? What is the blueprint of what we can change? Some of it we can, other of it, luckily with the human brain, is very plastic. We can change our brains quite a bit. But we're likely to find in the next 10 to 15 years, or at least see more confidently, that the changes specifically in receptors, neurotransmitters themselves, and reuptake of these and recycling of these neurotransmitters can control a lot of our emotional response. Now, the environment dictates a lot of that too, but it's very cool to consider what framework your brain is born with, or that of your patients or clients or anything like that, that they experience the world in a very different way. So yeah, when you have a little less serotonin, you tend to be a little more risk-taking, for example, and maybe less able to handle certain situations. So this lab is going to kind of engage this learning model, which is constructivism, which is you're going to go do stuff, you're going to make mistakes, and you're going to learn, and you can learn from those. So we'll talk more about this in lab, but it's kind of a cool hands-on thing. So awesome. That should do it. And thank you.